Professor Tony Ginn completed his undergraduate medical and anesthesia training in New Zealand, then moved to Hong Kong, where he rose to professor at the Chinese University of Hong Kong Prince of Wales Hospital. He also served as chairman of anesthesia departments in both Christchurch, New Zealand, and Hong Kong, as section editor for anesthesia and analgesia, and is a former ISAP president. Tony's clinical research areas are broad, including perioperative outcome studies of drugs and pharmacogenetics related to pain, postoperative delirium, myocardial injury, obstetric outcomes, and wound infection. Dr. Ginn's talk is titled Brain-Derived Neurotrophic Factor Polymorphisms are associated with chronic post-surgical pain, suggesting potential drug targets. So thank you, Dr. Ginn. Professor.
So, hey, good afternoon. Thank you for the introduction, Don, and thank you for the opportunity for me to present one of the research areas of our preclinical group, namely pain mechanisms, and particularly our BDNF work. So I've got no disclosures except that the work has been funded by a variety of funding bodies in Hong Kong and Australia. So the outline of the talk is I'll talk briefly about the clinical problem of chronic post-surgical pain, which I think is quite important for us as we actually have a role potentially in its prevention, comment on some of the genetic influences on pain and translational research, and then give a summary of our current research on chronic post-surgical pain. The problem isn't new. Famous people, Henrik Kellett, Charles Jensen, Clifford Wolf, over 10 years ago wrote this review in Lancet, essentially saying that persistent pain is very common. The thing is that it wasn't really recognized because anesthesiologists, unless they were pain physicians, didn't follow the patients up. And surgeons were very focused probably on just their surgical specialty and didn't realize that it was quite prevalent in all the other surgical specialties and didn't really have the ability to follow essentially everyone up. And I think as anesthesiologists in preoperative medicine, we actually have this opportunity. And it's interesting that even back then they thought that one of the risk factors for developing persistent pain was actually the intensity of acute post-operative pain. And our acronym organization, although they're a bit bigger than we are, IASP, made last year actually the Global Year Against Pain After Surgery and did quite a variety of promotional activities to publicize this problem, including really the definitions, because without a proper definition, the epidemiological research can have a sort of uncertain base. So chronic post-surgical pain, or sometimes persistent post-surgical pain, is pain that develops or increases in intensity after a surgical procedure, which has essentially persists beyond the healing process, which is taken really as 3 months duration. It may be actually a continuation of acute post-operative pain, or actually might occur after an asymptomatic period. And it's usually localized to the surgical field or a related area of innervation. And you've excluded other causes such as recurrence of the disease. And in the next ICD-11, the National Classification of Diseases, there will actually be a definition which should make database and epidemiological research a bit easier in the future. And as we heard from Tony, the incidence of this can be surprisingly high after some very minor procedures. And surprisingly, despite the advances in surgical techniques, the minimally invasive surgery, the incidence of chronic post-surgical pain doesn't seem to have really diminished with those minimal invasive procedures. And if we look at the predictive risk factors, again, there's many studies out there, and all of them come up with a variety of predictive risk factors, but very common ones include young age, sex, psychological factors, those that really have a stress and anxiety personality, depression, or catastrophizing behavior at increased risk of persistent post-surgical pain. The thing is, most of these factors really aren't changeable, but two which are essentially preoperative pain, and we heard last year how Duke has a essentially pain clinic, and I think Tony alluded to that today, that people with severe preoperative pain may actually be better to be brought in and manage their pain, reduce their opioids, to try and give them a better post-operative outcome. And similarly, severe acute post-operative pain is something that we as anesthesiologists can manage. And if you look at this large survey in Europe published a few years ago in 21 hospitals, they found that if you had increased amount of time with severe pain on day one, you had an increased possibility of chronic post-surgical pain. So for every 10% increase in the time that you spend in severe pain on the first day, you had a 30% increase in the instance of chronic post-surgical pain at 12 months. Now, pain you probably know is relatively complex. This slide is just really to show that in terms of pain mechanisms, you can look at all the way from the periphery to the spinal cord to the brain in terms of potential pathways. And this slide is every little box is some area that you could potentially interact with chronic pain pathway. Whether you want to look at leukocytes and cytokines in the periphery, some initial receptors and nerve endings. You want to go to the spinal cord and look at your GABA, etc., and all those. You want to look at your glial cells. Here it's nice that BDNF, TRK-B signaling is here. Or you want to look at the brain, look at your reward networks and things. There's lots of ways that people try to approach the problem of chronic pain. And of course, when you think each of these boxes represents some sort of pathway, you can imagine that each of these boxes, there are multiple targets. And again, if you look at this recent you're not expected to read this, but these are all the sort of potential targets for analgesics which have been looked at recently or currently. And so, when you go from ion channels, G protein receptors, neurotrophins, cytokines, enzymes, epigenetic mechanisms, cells, there's a lot of potential targets out there. But I'm sure you also know that really the history of pain research has actually been very poor. I know that earlier they were talking about how we haven't got any new anesthetic agents, and a few of them have failed recently. Well, the sort of track record of pain research is even worse. There's been hundreds of sort of failed pain trials. And I think part of that is because we don't really understand, and it's very complex, the development of or transition from acute to chronic pain. There's inflammatory pain, neuropathic pain, which share some similarities but also some differences. And there's no doubt that currently for chronic pain, the treatment options that we have are still pretty suboptimal with adverse effects here. Essentially, it's opioids with its known effects. You've got your gabapentinoids with its sedative effects. And there's really not been a great, a sort of innovative analgesic drug. We're just repackaging our opioids and reusing lignocaine and ketamine. And as I say, there's been a lot of failure of translational research. Now, there's always been interest in essentially, what about the genetics? And certainly it's estimated that, of course, you've got very similar circumstances, but not everyone with the same surgery and the same pre-mortem conditions develops chronic post-surgical pain. So the estimation is that at least half of the variance is related to genetics. And of course, this was a very sort of, I suppose, optimistic editorial in the Canadian Journal, which said that once we work out the genetics, you can do some personalised prevention, diagnosis and treatment. But we all know that the promise of personalised medicine hasn't really come about in most of medicine. And similarly, a lot of people were interested in polymorphisms of the mu-opioid receptor some years ago, again, which really haven't gone anywhere in terms of something interesting, therapeutic. And this recent article published in Anesthesiology from a Spanish group looked at a large number of patients, looked at chronic post-surgical pain at 4 months, and essentially found no significant genetic differences in 90 markers. So that wasn't very encouraging. Nevertheless, we still had this interest. And our idea was really not from bench to bedside, failure of translational research, but from bedside to bench. What we wanted to do was discover some genetic influences on persistent post-surgical pain in humans. Can we replicate that in an animal model? Once we replicate that in an animal model, then we do a variety of essentially cellular and in vivo experiments to try and determine the mechanism, and then find a target for the intervention, demonstrate that the therapy at least is effective in a preclinical model. And again, once you get to that stage, hopefully we'll go to a clinical trial. And so we have an ongoing persistent pain after surgery study, relatively simple. It's easy to recruit adult patients undergoing surgery. And essentially, you look at their clinical data, you do all your quality of life, depression, etc. scales, and you follow them up 3 months and 12 months. Essentially, you work out which of them have chronic post-surgical pain and which do not. At the same time, we've taken venous blood samples from them and do a targeted genotyping. We've been actually currently targeting about 700 different SNPs and about 20-something pain-related genes. And of course, you've got two groups, one with chronic post-surgical pain, one without, and you just see which are the significant differences in SNPs between them. And this is one of our early attempts at this work, kindly accepted by Mike a few years ago with an accompanying editorial, where we found that patients with a polymorphism of cathepsin G had a lower risk of chronic pain. Now, cathepsin G is a protease that regulates inflammation by action on immune cells and production cytokines. So this was essentially the human phenotype. In terms of having an animal phenotype, what we did, I think Howard alluded to this, we did a lot of essentially animal experiments looking at essentially inflammatory pain, and this is the classic complete Freund's adjuvant. You inject essentially some dried and activated mycobacterium with oil into the pore, and with saline after 7 days, I don't know if you can see, you don't get much inflammation, but it develops in the pore of the rat. And after you get this inflammation, you then test it typically for mechanical allodynia with von Frey hairs or thermal hyperalgesia with Hargreaves method. So it's reasonably standard in terms of animal research and in terms of chronic pain. And so what we found, if you look at the first slide, which we published, is that typically if you have a saline injection, you don't get any mechanical allodynia. Oh, sorry, this is thermal hyperalgesia. So if you get the thermal hyperalgesia, you'll get a very short time before they withdraw their pore. And if you actually give a cathepsin G inhibitor, you can actually attenuate some of that hyperalgesia. Even if you give it after the treatment, you can still attenuate some of the hyperalgesia. And so we did a number of studies reported in that paper showing that the cathepsin G was upregulated, had some effect on astrocytes. The only thing was that it really was unclear what role cathepsin G had as a SNP. And without doing the mechanism, it wasn't going to be very easy to work out how to address the sort of mechanistic pathway. So essentially we gave up on essentially trying to explore that further. However, again, what Evan kindly accepted earlier this year was our research with BDNF. And if we look at all the hit genes you get from BDNF, we actually had about something like, of the initial 700 and whatever, about 100 of them had a very low frequency, so we ended up about 600 with a frequency of more than 5%. And we had about 40 genes, some of which increased the chance of chronic pain, some decreased the chance of chronic pain. But the one that really stood out was BDNF. So even after you do a Bonferroni correction of P less than 0.05 divided by 600, the BDNF was still significant. And so in terms of the clinical part, this just goes through how we had the patients for the study. We had a discovery cohort, and as well as the discovery cohort, we actually did a separate validation cohort to show that the BDNF was associated with a lower risk of pain. And the conclusion really was that if you had allele A of this polymorphism, you had a lower risk of chronic post-surgical pain. And the odds ratios were quite significant actually, 0.62, 0.57 in the discovery and validation cohorts. It's actually quite a big difference in your risk of chronic pain. And if you look at the population attributable rate, 9% was higher than any other clinical risk factors in terms of predicting clinical pain. Now, BDNF is a secretory protein that regulates synaptic plasticity. We then went on and showed that in a mouse model, if you knock in this MET, then you can actually, in an incisional model, decrease the amount of mechanical allodynia that you get. So that was quite interesting. We actually have, we showed that in an animal model, the same polymorphism has a similar effect in terms of reducing pain. So if you look at the recent literature on BDNF, it's actually well known and actually probably well studied in terms of its relationship with pain. And BDNF, one of the neurotrophins, is a protein secreted by actually many different cells, not just neuronal cells, which stimulates and controls neurogenesis. It's actually important also for long-term memory. It's a neurotransmitter, and certainly it's probably well accepted that the BDNF from your microglia triggers your central sensitization at your spinal cord level. It binds to TrkB and P75 receptors, and there's multiple animal models essentially having antagonists of BDNF, etc., which over a different variety of animal inflammatory models, which show that if you block the BDNF, you decrease the pain. Unfortunately, because BDNF is quite ubiquitous throughout the body, and again, in terms of its name, brain-derived neurotrophic factor, if you take out the BDNF completely, it's really not compatible with survival. If you block the BDNF, actually you get depression and disturbed memory. And so pretty much, you need to preferentially block or target the BDNF, probably at the spinal cord and at the peripheral level rather than centrally at the brain. And if you look at this polymorphism, the Val66Met, it's in the pro-domain of actually BDNF, and it's unique to humans, and probably 20 to 30% of humans are heterozygous for the polymorphism. We presume that no one here has this because it's associated with a reduction in hippocampal tissue. It's more common in those with a learning, memory, or anxiety disorder, and actually it's more common in those with neurodegenerative diseases such as Parkinson's and Alzheimer's. Nevertheless, it is compatible with life. And if we look at actually BDNF, so what happens with BDNF is that you have the BDNF gene, which makes your sort of precursor pre-pro-BDNF, which at the Golgi network becomes pro-BDNF, and there's actually two pathways, a constitutive pathway, which releases steadily pro-BDNF and BDNF, and a regulated pathway, which puts BDNF into dense core vesicles. And it's actually this release upon a stimulation from the core vesicles, which seems to be responsible for the central sensitization. So immediately if you've got two pathways, and this pathway with a dense core vesicles is responsible for the pain, then maybe you can differentially affect the two pathways. And it's reasonably complicated in terms of the molecular biology, but certainly the mature BDNF, the pro-BDNF, all have slightly different actions on different receptors. But it was certainly been shown that if you have the Val-Met polymorphism, you actually affect the binding of a protein called sortilin. I said that the BDNF goes to two pathways. There's actually this protein sortilin which determines whether it goes to one pathway or the other. And this work from Cornell essentially found that you've got this protein called sortilin. It actually controls trafficking of a variety of neurotrophins and peptides. And what it does is it binds with the prodomain of BDNF to sort the BDNF to the regulated pathway. And by some knockouts of a variety of amino acids, these workers found that the interaction between the pro-BDNF and the sortilin occurs in a 44 to 102 amino acid sort of span. Quite a large span, but that was what they did. And if you interfere with this pathway, and you can see that the Val66Met falls within this amino acid sequence, you actually have less BDNF in the secretory vesicles for regulated pathway. And if that happens, then you're going to have less pain, less BDNF released to cause you your pain hypersensitivity. So this was really, I suppose, what I'm presenting here is work that's currently under review. So I'd be appreciated if you sort of keep this within the room. But anyway, in terms of our theory, you've got your pro-BDNF here. The sortilin in the green binds to the pro-domain. And normally what happens is that the sortilin binds to the pro-domain and packs the BDNF in the vesicles, which is waiting to be released to cause pain hypersensitivity. We thought that if you could block this interaction between the sortilin and the pro-domain with, in our case, a blocking peptide, then you wouldn't get any BDNF in these vesicles, you wouldn't get the release of BDNF, and you wouldn't get your pain hypersensitivity. So this, the next series of work that we're having is trying to establish essentially this pathway. So if we start right from the beginning, can we show that the BDNF and the sortilin is co-localized? Actually, they interact. And so this is some pretty standard immunofluorescence. You probably can't see, but essentially the green is going to be the BDNF, the red's the sortilin, and when they overlap, it becomes nicely orange, just so they're co-localized. You can also show, if you have the complete Freud's adjuvant pain model, that you increase the expression of BDNF in the ipsilateral dorsal spinal horn, and similarly, this Western blot shows that you get also increased sortilin in the dorsal spinal horn. So if you have this inflammatory pain model, you increase both BDNF and sortilin in the dorsal horn. Then we can take some adeno-associated virus, short hairpin RNA, and inject it into the L4 dorsal root ganglion of some mice. We knock down the sortilin by about 50%, and not that convincing, but you can actually reduce the mechanical allodynia with that. But I say we didn't knock down the sortilin that much, only about 50%. So once we've done that, and we're happy that the sortilin and the BDNF actually have an interaction, we then go on to try and work out, well, where is this interaction? The previous work had shown that the interaction was somewhere between amino acids 44 and 103. But that's quite a big span. So what we did is we cloned the human BDNF and the sortilin, and we had some essentially mutant constructs where we just deleted essentially 20 amino acid sequences, overlapping 20 amino acid sequences. And then, so you can see here, here's the full BDNF with the full amino acid sequence. What served as our control was the earlier people that had knocked out this large area of BDNF. So we knock out smaller bits of BDNF, and then by a variety of co-immunoprecipitation and biomolecular fluorescence complementation assays, try and work out, amongst that large area, which area is more likely to contain the interaction site. So if we look at the Western blot for the co-immunoprecipitation, it looks like area 4 here has less interaction between sortilin and BDNF. And that's really, number 4 is 71 to 90. But if we look at the biomolecular fluorescence complementation assay, this is when you take a venous fluorophore, it's a dimer. You stick half on the BDNF, half on the sortilin. And if they join together, they fluoresce. And so here we can see there's the control one that doesn't fluoresce, and both 4 and 5 don't fluoresce, as we can see here. So 4 and 5 are more the 71 to 100 range. And again, you can sort of look at the expression of BDNF in the supernatant of the cells. And again, both 1, 4, and 5 variants don't produce any BDNF in the supernatant. So in some ways that narrows our, our sort of area for interaction to somewhere between 71 and 100, which is better than 40 to 100 it was before. So once we do that, we then have to work out how to block it. So we construct a variety of blocking peptides. So we create some overlapping 10 amino acid peptides. And then you do further co-immunoprecipitation experiments with each of these different peptides and work out which peptide essentially blocks the interaction. Because the peptides don't penetrate the cell, we have to tag it with an HIV TAT, so by endocytosis you can get this peptide into the cells. And the summary of all that is that we had one particular peptide, 89 to 98, which had essentially the best at blocking the interaction between BDNF and sortilin. So once we have this blocking peptide of 10 amino acids, we then go on with further experiments where we have the blocking peptide, and then as a control we scramble the amino acids in different sequence, and again repeat all those immunoprecipitation experiments. As well as the cellular models, we then have to go on to do the in vivo models. So again, using the rat complete Freud's adjuvant model and the capsaicin challenge, we intrathecally inject the blocking peptide to see the effect of that on the allodynia and hyperalgesia. We look at downstream signaling. We know that BDNF binds the receptor and activates the ERK pathway, so we look at the concentration of phosphorylated ERK. And again, we also, instead of intrathecal injection, we also try to do intravenous injection of the blocking peptide. And here, well, this is, I suppose, a really quick summary. You've had all this before in terms of the design of blocking peptide. So these ones on the left, immunofluorescence studies, essentially showed that if you have the blocking peptide, you can decrease the amount of phosphorylated ERK. And again, these two down here are the in vivo models of mechanical allodynia and thermal hyperalgesia. Not a significant difference, but you could say not too it's significant. I'm sure we could probably refine the blocking peptide a bit more. Anyway, I suppose the summary of all this is that normally you have your pro-BDNF, interacts with your sortilin, packs your BDNF into these dense vehicles, gets released, you get downstream signaling and you get pain. If we block it with this peptide, you prevent the vesicles having this BDNF, and then you prevent the hypersensitivity and pain. And so that's a summary of the work that's being reviewed at the moment. And certainly in terms of pathways, this is a relatively interesting pathway. There are many other people who are looking at this pathway in terms of new analgesics. A group from, I think UCH, managed to actually delete BDNF selectively from sensory neurons and show that that's important for the transition from acute to chronic pain. As I say, the BDNF is reasonably complicated because both the pro-BDNF and the BDNF have actions on receptors. And so another group, Luo from China, are looking at sort of peripheral pro-BDNF and modulating that as in terms of chronic pain. And finally, FANZCA have published that they have a variety of peripherally restricted track kinase inhibitors, which they, I think, have got to phase 2 for as terms of a new analgesic. So in terms of the whole BDNF track pathway, this seems to be one of the areas where there might be some promise. And I say, certainly with our studies, we've shown that at least you have the human phenotype already, so hopefully that there should be some translational sort of success at the end if we can work out all the different mechanisms. So, in summary, I say that chronic post-surgical pain is still a significant problem. There's many risk factors, but most of them are not amenable to change. We really can't do gene therapy at the moment, but at least we can do the genetic study. But I think the genetic study not only has to identify that there is a difference, but really to investigate the mechanism and look at the pathway in detail to see if there's some translational potential by finding some new target. And certainly as anesthesiologists right now, we can actually do something to influence the development of chronic post-surgical pain in our patients by treating their acute post-operative pain, although this hasn't actually been confirmed in a sort of a clinical trial yet. And certainly I think those of us that do many multi-center clinical trials, it's probably quite useful to add a follow-up of chronic post-surgical pain if we can. We're often following these patients up for a long time anyway. And it's relatively easy to click this, and that I think will improve our understanding of chronic pain quite a lot more. Yeah, so this is my research group, thank you. Heidi Ho is a PhD student that did most of this. She's now off to Oxford to do some work with Irene Tracy with the functional MRI on pain. And so thank you.